Aliana, and I'm a Master's of Science candidate at the University of Guelph. I'm excited to be here with OE3C today to present my research on methods of enhancing selective fish passage. If you take a look around you, you'll likely see many barriers to animal movements. These barriers can be either naturally occurring or from anthropogenic activity, and they fragment the connectivity of habitats that animals use. Habitat fragmentation is where organisms' movement is limited, which prevents them from accessing habitats that can be used for feeding, breeding, and rearing offspring. If you took a look at the images on my screen, you can see some examples of barriers that an animal might encounter. In terrestrial habitats, roads and mountain ranges can limit movement. In aquatic habitats, dams and waterfalls can inhibit movement between habitats. Now, you might be sitting, listening to my talk and thinking, if animals naturally encounter barriers, why are we even listening to this presentation? My answer to your question is that the number of anthropogenic barriers or human-made barriers that animals encounter is rapidly increasing. In fact, global estimates suggest that 60% of rivers are impounded by at least one barrier. This is a major concern for ecosystem managers because it can have severe consequences for biodiversity. By limiting the movement between habitats, animals can be restricted to a poor quality habitat, which can limit growth and reproduction. These populations are then more susceptible to environmental and demographic stochasticity, which can lead to reduced breeding success, reduced life history diversity, and ultimately a loss in biodiversity. Now, to visualize how impacted our aquatic habitats are by barriers, I wanted to show you this image from a publication by Janikowski-Hartley and colleagues developed in 2013. Across the Great Lakes, they mapped the distribution of in-stream barriers, which is shown on the left in red, and road crossings, which is shown on the right in blue. Each point represents a single barrier. So specifically, I'd like to draw your attention to the top map that shows in-stream barriers in red, because that's going to be the focus of my talk today. While these points might appear sparse, the authors estimate that there are approximately 7,901 dams on tributaries of the Great Lakes. As we know that barriers fragment habitat connectivity and can have a negative impact on biodiversity, it makes sense that many fishery managers are arguing for the removal of these barriers. Now, as positive as this sounds, because the goal is to restore biodiversity, there are some challenges that are posed by restoring habitat connectivity. While reopening habitats means desirable fishes can access better spawning habitats, better feeding habitats, or better shelter from predators, an unintended consequence of restoring habitat connectivity is that invasive and predatory species can also disperse into these habitats. The introduction of these invasive species can further threaten aquatic biodiversity by introducing diseases, increasing interspecific competition, and increasing predation. For some endangered species, barriers have been built for the purpose of protecting them from predation. Removing these barriers can ultimately result in the extirpation of these species. So this has been termed the connectivity conundrum, where the maintenance of barriers can be detrimental for the persistence of desirable populations, but the restoration of connectivity also threatens these populations by allowing invasive species to spread. This makes it challenging for fishery managers who have to weigh the risks of invasive species dispersing and proliferating against the loss of desirable native and recreational species. Selective connectivity has been proposed as a potential method of resolving the connectivity conundrum. Selective connectivity is where the barrier is used to selectively fragment the habitat for undesirable species, while habitat connectivity is restored for desirable species. Sea lamprey are one such species whose populations are controlled by selective connectivity. Sea lamprey are invasive to the Great Lakes and parasitize large, economically valuable fish and are used in both recreation and fisheries. Adult sea lamprey leave lakes in the spring to migrate upstream to spawn, and barriers are used to block their spawning migration to prevent their population from further growing. So this trap and sort fishway shows how we can selectively fragment the habitat to control sea lamprey. So this is a two-chamber vertical slot fishway that's constructed adjacent to an in-stream barrier here, and it uses funnels here and here to sort fishes by size. I'm going to show you how the fishway can be used to selectively restore habitat connectivity. The white sucker will enter the fishway as it migrates upstream against the flow of water to spawn. It can pass through the large funnel, but cannot enter through the small funnel, and it becomes trapped in what we call the downstream chamber. So the sea lamprey, on the other hand, can pass through both funnels and is then trapped in the upstream chamber. Fishway technicians then manually translocate the desirable species, so our white sucker, from the downstream chamber to above the barrier so that it can continue its spawning migration. Sea lamprey are removed from the fishway and euthanized. Side-selective sorting, however, doesn't stop small-bodied fishes from entering the upstream chamber, which means extra sorting for fishway technicians, and this endeavor becomes very costly and time-consuming, which prevents it from being implemented on a larger scale. For selective connectivity to be more widely implemented, we need to find ways to reduce manual sorting. So one way of doing this would be to facilitate what's known as passive sorting, where fish sort themselves. To induce passive sorting, we require a stimulus to induce two different responses, one response in sea lamprey and one in desirable fish. River discharge, it provides migratory cues to fishes and induces rheotaxis, where fish orient themselves to the direction of flow. Sea lamprey are highly rheophilic, meaning that they are attracted to high river discharge. Studies of juvenile salmonids and whitesuckers have found that intermediate flows are more attractive and that fast and complex flows are avoided. Additionally, Pratt and colleagues in 2009 showed that higher discharge through the fishway encourages fish to enter and remain in the fishway, but fish were observed exiting the fishway when discharge through the fishway was too low. So given that river discharge induces two different responses into desirable fishes and invasive species, or the invasive sea lamprey, we could potentially exploit this stimulus for passive sorting. So we predict that if moderately high discharge through the fishway encourages fish to enter and remain in the fishway, then large desirable fish will be held in the lower chamber by the size-selective funnels, and the narrower sea lamprey will continue to the upper chamber, facilitating passive sorting. Alternatively, if lower discharge through the fishway does not encourage fishes to enter and remain in the fishway, then desirable fishes will exit the lower chamber and sea lamprey will not be encouraged to continue to the upper chamber, which undoes passive sorting. So we tested this in the summer of 2019 in Sault Ste Marie at the Big Carp River fishway. This was led by a team. This was led by Gail Bravener and Tom Pratt at the Big Carp River fishway located on the tributary of Lake Superior. Experiments were run from May 23rd until June 28th during the sea lamprey spawning run. The experiments were run in 2-day strata, where one night had high relative discharge through the fishway and the other night had low relative discharge through the fishway. High and low discharge days were randomly assigned within 2-day stratum to make a total of 18 strata. On each day, the total counts of sea lamprey and desirable fish species were recorded for each chamber, and desirable fishes were identified to the species level. Their total lengths were also recorded. If no sea lamprey or desirable fish were captured from the fishway, that stratum was excluded from analysis. Now, for us to quantify the effectiveness of passive sorting, we actually took a model that was used in the recycling industry. In 2010, Wolf and colleagues developed models to determine the effectiveness of sorting a binary material mixture, or a mixture of two materials. So the schematic on this slide shows how that mixture, that the sorting of those mixtures can be applied to the world of passive sorting. So we have an input stream on the left of two materials, so our desirable fishes and then our sea lamprey, and then they undergo a sorting mechanism. So in our case, this is either high or low discharge. That creates a primary stream of the target materials here, and the non-target materials, which are desirable fishes. For our purposes, the model evaluates the sorting of desirable fishes into one chamber and sea lamprey, which are our target material, into a separate chamber. So from this model, we can get two parameters. R, which is the recovery of the non-target material or desirable fishes in the downstream chamber, and Q, which is the recovery of the target sea lamprey in the upstream chamber. In this model, we evaluate the effectiveness of sorting by plotting the recovery of desirable fishes in the downstream chamber against the recovery of sea lamprey in the upstream chamber. So if we look at the plot that's developed by Wolf and colleagues, highly effective sorting results in all materials being recovered with R and Q values being 1, which is in that top right corner. So in our experiments, we're expecting that under high discharge conditions, it's highly effective at sorting sea lamprey and desirable fishes into their respective chambers, so the recovery should be 1 for both. When you're having low discharge conditions, we expect that the sorting of desirable fishes and sea lamprey is low, so we'd expect our points to be in this left corner. What we actually saw, though, was that the sorting of sea lamprey is highly effective, where Q was 1 for all strata. And so the sorting of our desirable fishes, however, was very low, with almost all strata being less than 25% of desirable fishes being recovered from the downstream chamber. Now, we evaluated the effectiveness of discharge on sorting of desirable fishes using a Bayesian binomial generalized linear mixed effects model using JAGS and R. The discharge treatment was determined not to have a significant effect on sorting, with the parameter estimate being negative 0.01 and the 95% credible interval overlapping with 0. A similar analysis was not performed for sea lamprey because there was no variability in sorting, as it was 100% for all strata. So our results indicate that while discharge may be effective at sorting sea lamprey, it's not effective at sorting desirable fishes. An unanticipated yet interesting result that we found was that despite discharge being ineffective at sorting, there were differences in the total catches of fish at the traps. The median catch on nights of low discharge was higher than on nights of high discharge for desirable fishes, and overall there were more desirable fishes found in the upstream chamber. The median capture of sea lamprey, however, was higher on nights of high discharge than on nights of low discharge, and all sea lamprey were captured in the upstream chamber. So these findings tell us two things. The first is that we can potentially sort fishes prior to entry into the trap using river discharge, as desirable fishes tend to be more present on nights of low discharge than on nights of high discharge. And that the problem with sorting isn't sea lamprey. It's those small-bodied desirable fishes that are able to get into that upstream chamber. While discharge-induced passive sorting doesn't appear to be effective, there are other methods of sorting that can be considered. Currently, I'm comparing the effectiveness of 4 different management options, which are shown in the boxes here from left to right. So we have manual sorting here, seasonal sorting, diurnal or daytime sorting, and varying durations of size selective sorting. The best management option will be determined by weighing the benefit of passive sorting against the risk of sea lamprey, the benefit of non-target passage against the risk of sea lamprey escapement, and the cost associated with the degree of manual sorting necessary for each option. Manual sorting is currently the standard since it's effective at ensuring that all fishes are in the trap are passed upstream. It has minimal risk of sea lamprey escapement, but it's a more costly option because it requires fish to be sorted by hand. Blocking the river on a seasonal basis minimizes the risk of sea lamprey escapement and has no cost associated with manual sorting, but it does block non-target fish movement. Blocking the trap at night and then opening it during the day when the sea lamprey are less active increases the risk of sea lamprey escapement, but minimizes costs associated with passive sorting and allows non-target fishes to pass. The last option is to utilize both manual and passive sorting, where passive sorting can be used for the first 7, 14, or 30 days of the spawning run, and manual sorting is used for the rest of the duration of the spawning run. Right now, I've started looking at evaluating the effectiveness of seasonal barriers using models that were developed by Velez-Espino in 2011. I plotted the capture frequency for 3 species of non-target fishes that represent the types of fish that are likely to encounter a barrier in a tributary when the tributary is blocked seasonally. Using data from 3 tributaries of the Great Lakes between 1995 and 2015, I compiled catches for 105 unique species. I selected species with more than 10 fish captured per year at each site for a total of 162 sample years. The species that I decided to select were the white sucker, the creek chub, and the common shiner. In addition to being found across the Great Lakes, these species also have different movement patterns, where white suckers are highly migratory, creek chub move within streams, and common shiners are relatively resident. For these species, I modeled the proportion of the catch expected on a certain day, in a certain year, for a certain tributary. I averaged those captures across tributaries in years to generate migration phenologies for each of the species I'm examining. The sea lamprey phenology is shown with the solid black line, and if we're trying to block 100% of sea lamprey migration using those seasonal barriers, we're likely to block the majority of non-target fishes. When I modeled the proportion of non-target fishes that were seasonally blocked, I found the proportion of non-target fishes blocked is almost 100% if the barrier operates for more than 60 days. Since traps operate for about 15 days, these results suggest that all fish will be blocked for normal seasonal barrier operation. These findings are in accordance with Dr. Velez-Espino's findings, which showed that even by reducing trap operation from 115 days to 75 days, 99% of desirable fish passage was block. Going forwards, I'll be using these models to incorporate 7, 14, or 30 days of passive sorting to see if there's any benefit to non-target passage while minimizing sea lamprey escapement. Additionally, I'm going to be evaluating the effectiveness of sorting during the day or diurnal sorting. So stay tuned for those results. Thank you for joining me today. I'd like to thank my advisor, Dr. Robert McLaughlin, and my committee members, Dr. Georgia Mason and Dr. Martin Coops, for their support on this project. I'd like to thank Gail Bravner, Tom Pratt, and Ryan Booth, as well as the DFO summer students for collecting this data. Thank you for showing up.